When glucose enters into the cell, it gets trapped by phosphorylation to glucose 6-phosphate, and glucose 6-phosphate has three pathways. The first one is glycolysis, the second one is that glucose can be used for glycogen synthesis, and the third one is hexose monophosphate shunt. The major function of hexose monophosphate shunt is production of NADPH molecules. Hexose monophosphate shunt is the major source of NADPH molecules in our body. Hexose monophosphate shunt occurs in the cytosol, and this process consists of two phases, oxidative phase and non-oxidative phase. The general picture of hexose monophosphate shunt is that when glucose enters into the cell, glucose is trapped by phosphorylation with formation of glucose 6-phosphate. Initially, glucose 6-phosphate is degraded in the oxidative phase of hexose monophosphate shunt. Oxidative phase results in production of two NADPH molecules, and the end product of oxidative phase is ribulose 5-phosphate. So basically the major function of oxidative phase is to provide NADPH molecules for the tissue. NADPH is an electron carrier that carries two electrons. So basically it's a reducing agent that serves as cofactor for several enzymes. And we have three major processes that require NADPH molecules. NADPH are required for antioxidant defense because NADPH are responsible for glutathione reduction. Also, NADPH are used in fatty acid synthesis and in cholesterol synthesis. As we see, NADPH is an absolutely vital agent. And we have tissues with the highest activity of hexose monophosphate shunt. It's lactate in mammary glands, liver and adrenal cortex. So basically it's a tissues with the highest activity of fatty acid synthesis and steroid synthesis. And also red blood cells, where NADPH molecules are required for antioxidant defense. Then the end product of oxidative phase, ribulose 5-phosphate, can be converted into ribose 5-phosphate, and ribose 5-phosphate is a substrate for nucleotide synthesis. Nucleotides, as we know, are required for synthesis of DNA and RNA molecules. If tissue do not require massive DNA synthesis at this particular moment, then ribose 5-phosphate is converted to glyceraldehyde 3-phosphate and fructose 6-phosphate. Both of them can be used in glycolysis or in gluconeogenesis. It all depends on energy state of the cell. If cell requires energy, then both these metabolites are used in glycolysis with energy production. If cell do not require energy, then they are used in gluconeogenesis that results in formation of glucose 6-phosphate. So basically, as we see, the most unique feature of hexose monophosphate shunt is production of NADPH molecule. And actually, hexose monophosphate shunt is the major source of NADPH molecules. Recall that we have two sources of NADPH. The major source is hexose monophosphate shunt, and the minor source is Malic enzyme. Malic enzyme uses NADP as cofactor and converts malate into pyruvate with release of NADPH molecule. Now let's discuss hexose monophosphate shunt in details. So once glucose enters into the cell, glucose gets phosphorylated to glucose 6-phosphate. So we have glucose 6-phosphate in the cytosol. To enter into hexose monophosphate shunt, glucose 6-phosphate is oxidized by the major enzyme in this pathway, glucose 6-phosphate dehydrogenase. Glucose 6-phosphate dehydrogenase uses NADP as coenzyme and converts glucose 6-phosphate into 6-phosphogluconate with simultaneous release of NADPH. Then 6-phosphogluconate is oxidized to ribulose 5-phosphate with release of NADPH. These two steps where glucose undergo oxidation with release of NADPH called oxidative phase of hexose monophosphate shunt. Because these reactions can occur only in one direction, this phase also called irreversible phase. And the most important is that oxidative phase results in production of two NADPH molecules. The non-oxidative phase begins with conversion of ribulose 5-phosphate. Half of ribulose 5-phosphate molecules are converted to oxyulose 5-phosphate and another part into ribose 5-phosphate. Then both these metabolites react with each other in reaction that is catalyzed by Transketolase enzyme with formation of heptulose 7-phosphate and glyceraldehyde 3-phosphate. And now these two metabolites react with each other. This reaction is catalyzed by Transaldolase enzyme with formation of erythrophosphate and fructose 6-phosphate. and Irriversible react with xylose 5-phosphate in reaction catalyzed by Transketolase with formation of glyceraldehyde 3-phosphate and fructose 6-phosphate. Important that Transketolase and Transaldolase enzymes catalyze all these reactions in both directions. And very high yield to know that Transketolase as coenzyme use thiamine pyrophosphate, which is vitamin B1. So basically it's a general picture how Hexose monophosphate shunt is organized. But we have to know that Hexose monophosphate shunt is connected to glycolysis and gluconeogenesis, and also Hexose monophosphate shunt provides the crucial substrate for nucleotide synthesis. First of all, Hexose monophosphate shunt is connected to glycolysis and gluconeogenesis. Recall that in glycolysis, initially glucose 6-phosphate is converted to fructose 6-phosphate, then phosphorylation occurs that results in production of fructose 1,6-bisphosphate, and then fructose 1,6-bisphosphate is cleaved into glyceraldehyde 3-phosphate and dehydroxyacetone phosphate. Then dehydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate, and glyceraldehyde 3-phosphate is oxidized to 1,3-bisphosphoglycerate. Then during glycolysis, 1,3-bisphosphoglycerate through 2 consecutive reactions is converted into phosphoenol pyruvate, and then phosphoenol pyruvate is converted into pyruvate. And pyruvate, as we know, is the final product of glycolysis, and most of pyruvate molecules are converted into acetyl-CoA that are used for energy production. In gluconeogenesis, organisms from pyruvate produce glucose, and basically glycolysis and gluconeogenesis have the same intermediate metabolites. But in gluconeogenesis, this process occurs in reverse direction, from pyruvate to glucose 6-phosphate. So we see that glyceraldehyde 3-phosphate and fructose 6-phosphate are the intermediate metabolites of both glycolysis, gluconeogenesis, and Hexose monophosphate shunt. In addition to this, glyceraldehyde 3-phosphate and fructose 6-phosphate are located in the cytosol. And all these reactions that use these metabolites in glycolysis, gluconeogenesis, and Hexose monophosphate shunt occur in the cytosol. So glyceraldehyde 3-phosphate and fructose 6-phosphate can be used in all these 3 pathways. It all depends on cellular demands. Also, ribose 5-phosphate is the intermediate metabolite that is required for synthesis of nucleotides. And nucleotides, as we know, are used in DNA and RNA synthesis. So Hexose monophosphate shunt by ribose 5-phosphate is connected to nucleotide synthesis. And now very important concept, because as we see all non-oxidative reactions can occur in both directions, this reversible feature permits to use glucose-derived metabolites by glycolysis, gluconeogenesis, and Hexose monophosphate shunt. It all depends on cellular demands. The major factor that determine by which pathway glucose 6-phosphate will be used is the ratio of NADP to NADPH. To explain this, recall that the major rate-limiting enzyme in Hexose monophosphate shunt is glucose 6-phosphate dehydrogenase. And glucose 6-phosphate dehydrogenase basically uptake glucose 6-phosphate into oxidative phase of Hexose monophosphate shunt. Oxidative phase results in production of NADPH molecules. Increase in NADPH, which is the product of oxidative pathway, allosterically inhibits this enzyme. So basically, if tissue have a lot of NADPH molecules, there is no point to generate additional NADPH molecules, so there is no need to use massive amount of glucose in oxidative part of Hexose monophosphate shunt. Better to use glucose for energy production in glycolysis, for example, or for nucleotide synthesis. And increase in NADP allosterically stimulates this enzyme, because if the amount of NADP increase, then logically that amount of NADPH is low. So basically tissue requires additional NADPH molecules that can be produced by oxidative phase of Hexose monophosphate shunt. So high NADP level stimulates glucose 6-phosphate dehydrogenase that takes glucose into oxidative phase, and during oxidative phase, NADP pluck out electrons from glucose, and this results in production of NADPH, thereby this causes increase in NADPH level inside the cell. So increase in NADP to NADPH ratio increases the activity of glucose 6-phosphate dehydrogenase, thereby stimulating Hexose monophosphate shunt, and decrease in NADP to NADPH ratio decrease the activity of glucose-6-phosphate dehydrogenase, thereby inhibiting Hexose monophosphate shunt. Another factors that determine the activity of glucose-6-phosphate dehydrogenase enzyme are ATP and ADP. Basically, in energy deficiency, tissue degrade ATP to ADP with release of energy. So this results in increase in ADP and decrease in ATP level. So if ADP level in tissue increase, this means that tissue requires additional energy. And in energy deficient state, generation of additional energy becomes the first priority. So high ADP level decreases the activity of glucose-6-phosphate dehydrogenase enzyme, and inhibition of Hexose monophosphate shunt basically leaves glucose for glycolysis, and the increase in amount of glucose that undergo glycolysis results in increase in energy production, thereby correcting energy deficiency. And obviously, in energy saturated state, ATP level in tissue is high, so cell do not require additional energy, and this permits to use glucose-6-phosphate for NADPH production that will provide NADPH molecules for fatty acid synthesis, and also this will provide ribose 5-phosphate for nucleotide synthesis. Because, for example, after the intake of high carbohydrate meal, a lot of glucose molecules income into the cell. Glucose undergo glycolysis that results in production of pyruvate molecules, and then pyruvate molecules are converted into acetyl-CoA, and in energy-saturated state, tissue do not require additional energy. In energy-saturated state, tissue prefer to store acetyl-CoA in form of fatty acids and triacylglycerols molecules. So cell activate fatty acid synthesis that converts numerous amount of acetyl-CoA molecules into their fatty acids. This process requires NADPH molecules that are generated in oxidative phase of Hexose monophosphate shunt, and then fatty acids bind to glycerol with formation of triacylglycerols that can be stored in tissues. So in such condition, during massive production of fatty acids, cell needs NADPH more than it needs pentose. So to generate more NADPH, ribose 5-phosphate that is used for nucleotide synthesis has to be converted back to glucose-6-phosphate that can be used again in oxidative phase of Hexose monophosphate shunt. So initially, ribose 5-phosphate is converted to glyceraldehyde 3-phosphate that subsequently is converted into fructose 6-phosphate, and fructose 6-phosphate is converted to glucose 6-phosphate that through oxidation by glucose 6-phosphate dehydrogenase enters into Hexose monophosphate shunt that produce NADPH molecules for fatty acid synthesis. Also, for example, when ribose 5-phosphate is produced in excess, Transketolase can shunt it into glycolysis for ATP production. And for example, when tissue regenerates, cells rapidly proliferate, and rapid proliferation requires massive DNA and RNA synthesis. And the substrates for the DNA and RNA synthesis are nucleotides. So in order to increase nucleotide synthesis, cell has to increase the amount of ribose 5-phosphate. And if oxidative pathway cannot produce enough ribose 5-phosphate, cell has to find the additional source of ribose 5-phosphate. So to cover the demand, Transaldolase and Transketolase enzymes convert fructose 6-phosphate and glyceraldehyde 3-phosphate from glycolysis to ribose 5-phosphate that can be used for nucleotide synthesis. So as we see, because Transaldolase and Transketolase enzymes catalyze reactions in both directions, non-oxidative pathway called the reversible pathway, and this feature provides extremely high mobility of glucose metabolites in the cytosol. So anytime Transketolase and Transaldolase enzymes can shunt metabolites from glycolysis into oxidative pathway or into nucleotide synthesis. And during energy deficiency, metabolites from Hexose monophosphate shunt can be also shunted into glycolysis that will provide additional energy for the tissue. So because all these glucose metabolites in the cytosol can be shunted into glycolysis, oxidative pathway or into nucleotide synthesis, this process called shunt, and Hexose monophosphate part just emphasizes the substrate from which all these metabolites are produced.